If you were to look at the brain of a 2-year-old macaque monkey, you'd see this fully mature, highly functioning organ. Like, it's efficient, it's capable.
Right, it's basically ready for the world.
Exactly, ready to go. But if you wanted to find that exact same level of genetic maturity in a human brain, you would have to wait until that human was, like, graduating from high school.
Yeah, 18 years. It's wild.
18 years. So today, we are unpacking one of the greatest mysteries in human biology. Basically, why is our ultimate evolutionary superpower tied to being so incredibly helpless for so long?
It is. It's a really profound paradox, I think, because usually, when we think of an advanced species, we just assume the biological mechanisms are faster or leaner.
Right, like more efficient.
Exactly, more efficient. But human cognition is the exact opposite. It's incredibly slow.
Okay, let's unpack this. For you listening, I want you to imagine 2 massive construction sites. On one side, you have our closest primate relative, the macaque monkey, and they're building a highly functional, solid office building.
Yeah, the scaffolding goes up, the workers pour the concrete, and boom, it's open for business.
Right, in just a few years. But on the other side, the human construction site is just lingering for decades. The scaffolding stays up, the workers are meticulously adding floors, then completely demolishing them, rerouting miles of electrical wire.
Taking just a staggering amount of time to finish.
Right, to finish this towering skyscraper of cognition.
Yeah.
And the source for our deep dive today gives us the actual blueprint for that skyscraper.
It really does. That visual perfectly captures what we're looking at today. We're diving into this groundbreaking paper that was recently published in Nature Neuroscience.
So it's a massive study, right?
Oh, huge. This research team managed to create a comprehensive single-cell spatiotemporal blueprint of the developing prefrontal cortex in both humans and macaques.
And just as a quick refresher, the prefrontal cortex, that's essentially the CEO of the brain, right?
Yeah, exactly. It handles our most complex cognitive behaviors. So personality expression, decision-making, moderating social behavior, language, all the heavy lifting.
Okay, so the mission of this deep dive is to basically read that master blueprint. We want to understand exactly how this incredibly protracted, slow construction timeline gives us our unique cognitive abilities.
But also explore the hidden costs.
Yeah.
Right. Because taking almost 2 decades to build a brain leaves us uniquely vulnerable to a whole host of neurodevelopmental conditions.
Yeah, we'll definitely get into that. But let's start with the sheer scale of this project. The paper notes they profiled over 1.8 million individual cells. I mean, how do you even begin to map something like that?
Well, the methodology here is really what makes this paper a landmark study. They analyzed 1.08 million human cells and about 737,000 macaque cells.
Wow.
And they didn't just count them, right? They used a multi-omics approach, which is it's basically a fancy way of saying they combined 3 separate cutting-edge technologies to interrogate these cells at the deepest possible level.
Okay, walk me through those 3 technologies. What are we actually measuring here?
Sure. So the first is called snRNA-seq, or single-nucleus RNA sequencing. If the DNA in a cell is the master instruction manual, the RNA represents the active work orders. So by sequencing the RNA inside a single nucleus, we can see exactly which genes are turned on and actively producing proteins at that exact millisecond.
Oh, gotcha. So it's like looking at the clipboard of every single construction worker on the site to see what specific task they're doing right now.
Precisely. That's a great analogy. Now, the second technology is SNATAC-seq, which measures chromatin accessibility.
Chromatin.
Yeah. So every cell in your body has the exact same DNA, right? What makes a neuron different from a heart cell is how that DNA is packaged. Chromatin is the material that spools and packages the DNA.
Right, okay.
So if the DNA is tightly coiled up, the cell can't read it. It's essentially padlocked shut.
So keeping with the site analogy, it's like having the master blueprint on site, but certain chapters are locked in a safe, while others are spread open on the table.
Exactly. You nailed it. SNATAC-seq tells us where those epigenetic switches are like, which parts of the genome are open and ready to be read.
Okay, so we've got the clipboards, we've got the open blueprints. What's the third one?
The third is spatial transcriptomics. This is essentially a 3D GPS tracker. The cortex has 6 distinct layers, and this technology allows the researchers to see the exact physical location of every single cell while it's expressing those genes.
Okay, so we know what the worker's doing, we know what instructions they have access to, and we know exactly what floor of the building they're standing on.
Exactly. And when the researchers took all of this 3D data and aligned the developmental timelines of humans and macaques, the results were genuinely jarring.
Yeah, I saw this in the paper. They found that a human baby at birth, so year 0, has a prefrontal cortex that aligns with a macaque that hasn't even been born yet, right?
Yeah, it aligns to a macaque at embryonic day 110.
That is wild.
The data bears it out. Biologically speaking, the human prefrontal cortex is just vastly undercooked at birth, and the divergence only gets more extreme from there.
Right, because when they profiled a macaque that was 2 to 4 years old, which, by the way, is a mature, self-sufficient monkey, its brain profile matched a human who is 18 years old.
Yep, an 18-year-old high school senior.
This brings to mind this microwave versus slow cooker analogy. The macaque brain is like a microwave meal. It's highly structured, you pop it in, and it's ready to function almost immediately.
Right.
But the human brain is in the slow cooker for nearly 2 decades.
I have to ask, from an evolutionary standpoint, why are human babies so famously helpless?
It's a fair question.
Like, we can't walk, we can't feed ourselves, we're entirely dependent. Is being born prematurely, basically, compared to a monkey, actually a strategic advantage?
No, absolutely. If we connect this to the bigger picture, that helplessness is absolutely a feature, not a bug. By stretching this developmental window over 18 years, the human brain isn't forced to rely purely on hardwired genetics.
Oh, I see. It remains incredibly plastic. This means the actual physical architecture of the brain can be sculpted by long-term environmental stimuli. Social interactions, complex language exposure, cultural learning, all of these elements become active ingredients.
So the slow cooker approach allows the environment to literally wire the hardware.
Exactly. It's brilliant when you think about it.
Okay, let's do a quick recap here for everyone listening. We're about 5 minutes in. So far, we've learned 3 main things. 1, scientists mapped over 1 million cells using multiomics.
Right.
2, human brains are basically raw and undercooked at birth compared to macaques. And 3, human brain development stretches over 18 years compared to a macaque's 4.
Perfect summary.
So for you listening, think about this. Have you ever considered that the sheer helplessness of human childhood is actually a highly evolved, intentional strategy for extended brain plasticity?
It really reframes how we view childhood, doesn't it? It's not just a period of waiting to grow up. It's an active, protracted phase of neurological engineering.
Which brings us perfectly into the actual mechanics of that engineering. We know the schedule is stretched out, but what exactly are these cellular workers doing during all those extra years?
Right, let's get into the mechanics. To track this, the researchers divided human development into specific chronological windows.
Okay, lay them out for me.
Window 1 is infancy, so 0 to 1 year old. Window the second is toddlerhood, ages 1 to 3. Window the third is preschool, ages 4 to 5. And window the fourth is childhood, ages 6 to 11.
Got it.
During window 1, that first year of life, the brain undergoes massive synaptogenesis. It is just furiously building connections or synapses between neurons.
Like laying down miles and miles of raw electrical wiring in the skyscraper.
Exactly. And we can track this chemically, which is so cool.
Yeah.
The researchers measured a presynaptic protein called VMP2.
VMP2.
You can think of VMP2 as the shipping department for neurotransmitters. It's the machinery that sends the chemical signal out of the neuron. In humans, VMP2 density is 4 times higher than in macaques during this window.
4 times? That's a huge difference.
Yeah. And they also measured PSD95, which is a postsynaptic protein. That acts like the receiving dock on the adjacent neuron catching those signals, and that was 2.5 times higher in humans.
Okay, so human infants are just flooded with these connection points. They are totally overwiring the building. But then, as I understand it, the blueprint calls for a pretty aggressive shift.
It does. Moving into windows 2 and 3, so the toddler and preschool years, the brain initiates a massive pruning phase.
Pruning, like cutting things back.
Right. The data shows a massive peak in the expression of a gene called CX3CL1.
CX3CL1, I'm going to need an analogy for that one.
Think of it as a biological demolition tag. This gene circulates through the brain and marks specific newly built synapses to be destroyed.
Wow, okay.
Furthermore, the researchers used a staining technique to look for a protein called caspase-3. Now, caspase-3 is the executioner enzyme for programmed cell death.
Executioner enzyme.
Yes. And they found a shocking 10% to 30% of cells in the human prefrontal cortex undergo programmed cell death during the toddler years.
Wait, hang on. I'm struggling to see the biological logic here.
I know, it sounds counterintuitive.
Yeah, I mean, you're telling me the human body spends a massive amount of metabolic energy in the first year of life, furiously pumping out VMP2 and PSD95, building billions of connections, only to send in, like, executioner enzymes to slaughter up to 30% of them a year later?
Yep.
Why not just build the right connections from the start? That seems horribly inefficient.
What's fascinating here is that it highlights how biological efficiency differs from, like, mechanical efficiency. The human brain overbuilds its wiring intentionally.
Intentional.
Yes. It creates a vast excess of synapses to capture all possible environmental input. Literally every sound, every visual pattern, every language phone the infant is exposed to.
Oh, I see.
Then, based on what the child actually experiences and practices, the brain ruthlessly destroys the connections that aren't being used.
Oh, wow. It's like pouring a giant block of marble to capture every possible shape, and then the toddler's environment is the chisel that carves away 30% of the stone to leave behind a custom-sculpted masterpiece.
It is an incredibly elegant way to conceptualize it. The pruning is what creates the precision. By clearing out the unused noise, the remaining circuits become faster, stronger, and completely specialized to that specific child's environment.
And do macaques do this too?
No, and here is the crucial species difference. Macaques simply do not show this same dramatic, protracted drop in synaptic density after birth. Their brains are much more rigidly hardwired from the start.
Which limits their ultimate cognitive flexibility.
Exactly.
Let's do another quick recap. We're at the 10-minute mark. For you listening, here's a quick mnemonic to remember this process. Build, prune, tune.
Oh, I like that.
Yeah, so build from 0 to 1 year, prune from 1 to 5 years, and tune from 6 to 11 years. So think about a toddler you know. Can you visualize their brain literally pruning away billions of connections right now to specialize in their specific environment?
It's pretty mind-blowing when you watch a toddler playing with blocks, knowing there's a microscopic demolition derby happening in their prefrontal cortex.
Absolutely. Okay, so this evolutionary trade-off is fascinating. We are essentially trading metabolic efficiency and, wasting energy building and destroying tissue in exchange for ultimate cognitive adaptability.
Right.
Let's look closer at the actual workforce executing this complex construction and demolition. The timeline is unique, but the paper also highlights that the cells themselves are distinctly human.
They really are. So the researchers identified 24 major cell types in the prefrontal cortex. And when they looked at which cells had the highest proportion of uniquely human genes, meaning genes operating differently than in macaques, one specific neuron jumped to the very top of the list.
Okay, what is it?
It's called the excitatory neuron L5-PT, which stands for layer 5 pyramidal tract.
Now, the cortex has 6 layers, right? So these are sitting deep down near the bottom. What makes layer 5 so special?
Layer 5 neurons are the output heavyweights. They consolidate all the processing from the upper layers and send signals deep into the brainstem and the spinal cord. And an astonishing 41.2% of the differentially expressed genes in this specific cell type were specific to humans.
41%. That's almost half the genes behaving completely differently.
Exactly. They also found entire subpopulations of neurons that are exclusive to humans. For example, there's one called the L2-IT-CUX2-CHN2 neuron.
Okay, that is quite the barcode.
Yeah, the nomenclature is pretty dense, I know. But the function is what really matters here. The IT stands for intratelencephalic, meaning these neurons communicate laterally across different hemispheres of the cortex.
Got it, like a crosstown communication wire.
Right. But what's truly unique about these human-exclusive neurons is that early in development, they highly express a gene called DAB1.
DAB1, and what does that do?
DAB1 makes these cells incredibly migratory. They are physically roaming around the 3D construction site to find their optimal positions.
That's crazy to picture, just cells wandering around the brain.
It is. And all of this complex wiring, this migration, is overseen by a master genetic switch called MEF2C. This is a transcription factor that specifically drives human synaptic signaling and memory formation.
Okay, but neurons aren't the only ones on site, right? The paper spends a tremendous amount of time analyzing glial cells, specifically astrocytes. And historically, we were taught that astrocytes were just brain glue, like just passive structural support to hold the neurons in place.
Well, yeah, the name glia literally comes from the Greek word for glue. But this paper firmly proves that astrocytes are actually the project managers of the brain. They actively regulate how synapses form, how they function, and when they get pruned.
Okay, the project managers, I like that.
And here we see another massive species difference. Humans maintain a huge pool of astrocyte progenitor cells, or APCs, long after birth.
And progenitor cells are basically stem cells.
Exactly. They're stem cells, waiting in reserve to turn into full astrocytes. In the postnatal human brain, these progenitors make up nearly 9% of all astrocytes.
Wow. And in macaque?
They almost entirely deplete them, down to less than 2%.
Okay, so how do humans manage to keep that workforce active and, proliferating for so long?
The data points to specific human-enriched genes, primarily CD44 and IGFBP5.
CD44 and IGFBP5, okay.
Yeah, these genes actively keep the progenitor cells in a proliferative state. And furthermore, the paper maps out how these astrocytes communicate with different layers of neurons using distinct ligand receptor pairs.
I picture that like a secret radio frequency. The astrocyte broadcasts a very specific chemical signal, the ligand, and only the neuron equipped with the matching receptor antenna can hear the instruction.
That is a perfect way to visualize it.
So if astrocytes are managing the construction site using these unique radio frequencies, and we have a vastly larger active pool of them, does this mean our higher glia to neuron ratio is the real secret to human intelligence, rather than just having more neurons?
The scientific community is certainly leaning heavily in that direction. This extended astrocyte proliferation perfectly aligns with our extended developmental windows for really complex behaviors, things like language acquisition and high-level abstract memory.
Right.
The neurons are the electrical wires, but the astrocytes are the master architects, actively managing where those wires go and how strongly they connect.
Okay, time for our 15-minute recap. So, summarizing this cellular workforce. 1. Humans have unique, highly migratory neuron subtypes. 2. MEF2C acts as a master regulator for memory genes. And 3. Human brains maintain a massive active workforce of astrocyte progenitors regulated by CD44 and IGFBP5 long after birth.
Spot on.
So for you listening, how does it change your view of intelligence to know that the support cells, the astrocytes, might actually be the master architects of human cognition? It totally shifts the paradigm away from just counting neurons, doesn't it?
It really is a symphony of cellular communication that macaques simply don't possess at the same scale or duration.
But of course, there is a catch. Because a construction project that takes 18 years to complete involves millions of highly specialized roaming workers and requires massive intentional demolition phases, well, it has a much higher statistical chance of going wrong.
Yeah. And this brings us to the cost of complexity. The researchers directly map how this protracted developmental timeline is linked to vulnerabilities for neurodevelopmental and neuropsychiatric disorders, conditions such as autism spectrum disorder, ADHD, and schizophrenia.
Now, it's worth pausing here because the conversation around neurodivergence is incredibly nuanced today, right? Encompassing everything from social models of disability to neurodiversity advocacy. So to be clear for you listening, we are looking strictly through the lens of this specific Nature paper and its biological mapping.
Right, absolutely.
So from a purely genetic and structural standpoint, how did the researchers link this slow cooker brain development to these vulnerabilities?
They focused on transcription factors, which we touched on earlier with MEF2C. Transcription factors are the master genetic switches that tell other genes when to turn on and off.
Okay.
The researchers found two specific families of transcription factors that showed massive developmental differences between humans and macaques. Let's look at them sequentially. First is the CUT family, specifically the genes CUX1 and CUX2.
CUX1 and CUX2. Got it.
These genes operate primarily in the upper layers of the cortex.
And what happens if those CUX genes mutate or just don't express correctly?
Well, extensive genetic mapping has strongly linked variations in the CUX genes to higher risks of schizophrenia and autism spectrum disorder.
Okay, and what was the second family?
The second is the fork family, specifically FOXP1 and FOXP2. These operate deeper down in the cortex. Now, FOXP2 in particular is really famous in evolutionary biology. Mutations in this gene are profoundly associated with severe speech and language disorders, as well as ASD.
Okay, but the researchers didn't just point to a statistical link, though, did they? They actually proved what these switches do using an ex vivo experiment. Can you explain how they managed to test this?
Oh, this part is phenomenal laboratory science. So ex vivo means out of the living. The researchers essentially grew living, cultured human cortical tissue in a petri dish. They kept a slice of human brain tissue alive and functioning in the lab.
Wow.
Then they used advanced genetic tools to selectively knock down or temporarily turn off these specific genes for 14 days.
Wait, really? So what happened to the neural construction site when those master switches were just turned off?
The architecture began to collapse. When they knocked down CUX12 or FOXP12, the affected neurons exhibited a drastic reduction in dendrite complexity.
Dendrites beam.
Dendrites are the tree-like branches extending from the neuron that reach out to receive signals from other cells. Think of them like the branches of a tree seeking sunlight.
Okay.
Without these specific transcription factors, the branches literally withered. The neurons could no longer form the complex, dense networks required for higher human cognition.
So the very genes that give us our advanced human capabilities, like the FOXP2 language gene, are the exact structural load-bearing pillars that, if disrupted, lead to these neurodevelopmental conditions.
Exactly. The researchers mapped this vulnerability specifically to the balance of the brain. You have deep-layer excitatory neurons, which push electrical signals forward, acting like the gas pedal.
The gas pedal, okay.
And you have a specific type of inhibitory cell called a parvalbumin, or PV neuron, which acts as the brake pedal. And astrocytes manage the balance between the two.
Ah, the excitatory-inhibitory imbalance model. I've heard of this.
Yes, exactly. If the deep-layer excitatory neurons and the PV inhibitory brakes don't develop in perfect sync over those protracted 18 years async that is heavily managed by those human-enriched transcription factors, the complex circuitry for spatial learning, language, and social processing can be permanently altered.
It really forces us to ask a difficult question. Is this vulnerability to psychiatric and neurodevelopmental conditions just the unavoidable tax we pay as a species for having such a long, complex, and specialized brain development?
Well, the scientific consensus drawn from this text heavily suggests yes. Because our higher cognition relies entirely on a protracted, nearly 2-decade window of neural plasticity, that same open window leaves the delicate balance of neural wiring exposed to genetic anomalies and environmental disruptions for a vastly longer period of time than any other primate.
Which perfectly ties the entire narrative of this deep dive together. We started by looking at a construction site, and what we've learned is that a 4-year-old human brain is not a miniature finished building.
Not at all.
It is a chaotic, hyperactive site. It is furiously building millions of connections and then ruthlessly pruning them away, guided by migratory human-specific neurons and overseen by a massive active workforce of astrocyte project managers.
Yeah.
The sheer length and fragility of this process explains exactly why early childhood environments are so incredibly critical. It fundamentally changes how we view learning and child development. I mean, the environment isn't just something happening to the child. It is literally the chisel writing the physical architecture of their brain.
It is the ultimate ongoing collaboration between our biological blueprint and our lived experience.
So as we wrap up, I want to leave you, our listener, with a final thought to mull over. We've established today that human cognitive supremacy relies entirely on a deeply protracted, slow-cooker childhood, where the brain takes nearly 2 decades to meticulously sculpt its skyscraper.
Right.
But today we live in an era of hyper-fast information. We are surrounded by infinite scrolling screens, algorithmic instant gratification, and intense societal pressure to accelerate learning and maturity in young children. So if our ultimate evolutionary superpower is our unique ability to take our absolute time to mature, what are the hidden biological costs of forcing a slow cooker brain to adapt to a microwave world? Think about that the next time you see a toddler taking their time to explore the world. Until next time, thanks for diving deep with us.